Welcome back to Oncology for the Inquisitive Mind, our second of a planned 3 episodes on ASCO. And unlike previous years, we are actually keeping to both time and schedule. Truly, miracles are real. I am joined by Josh, who is just as dapper as he was last night. I think I'm looking a bit more casual. But Josh, are you recovered and rearing to go for our second episode?
Michael, I just could not sleep last night. That is how excited I was to see you on screen to talk about all the incredible studies that came out of ASCO 2026.
And has absolutely nothing to do with the fact that you have 2 young kids.
Nothing at all, I promise. I lie. It's always the kids.
It's always the kids. So as mentioned before we launch into this our list of chosen subjects, this episode is going to be on agents for the future. So that's studies with novel agents or novel applications of old agents that we think show promise or meet an area of need, but we have a number of questions about them. And Josh, why don't you start folding everything into place with Origami 4, which is the first study that we have.
Sounds good. Agents of the future, my friend. Agents of the future.
As opposed to Agents of Mayhem, which is what the show is normally about.
It's just to mayhem.
Chaos.
Oncology.
Yes.
Yeah, chaos. So Origami-4. It's a head and neck cancer study, which is great because, again, really morbid disease, both early and late setting. Current first-line standard of care for metastatic head and neck squamous SCC is immunotherapy plus or minus platinum-based chemotherapy. And remember, folks, immunotherapy only came on in like the last, 2-3 years. Before that, it was just chemotherapy, and it wasn't fantastic. Treatment options after first-line are limited in efficacy, and usually you're looking for a trial or people are very unwell, and that includes the classic cetuximab, which has been around for donkey's years, with an objective response rate of about 24% and a median progression-free survival of 3.8 months. And look, some people definitely get longer than that, and some people get shorter, but overall, not fantastic. And prognosis is poor. With this cohort of patients, EGFR and MET mutations are frequently overexpressed. And back in the day, you could figure this out, and there's nothing you could do about it, but things are changing. With this cohort 1 study readout for Origami 4, which is evaluated amivantamab. The method was 102 patients, just in case you're curious. They were enrolled when you progressed after first line. They received amivantamab. I can't pronounce anything today, Michael. On cycle 1, day 1, and then 2,400 milligrams on cycle 1, day 8, and day 15 every 3 weeks. So a relatively intense regimen, and given subcutaneously. So that's always nice. Could do here, hospital in the home, just get them there if this ends up being something great. Patients could not have received prior anti-EGFR therapy before, and the primary endpoint was objective response rate, and secondary endpoint was duration of response, progression-free survival, etc. It's a small study. So the objective response rate was 47%. You had 4 patients that had complete response, 44 patients that had partial response, and 39 that had stable disease. I would give that a pretty solid round of applause in my book. Average responders, the median duration of response was 7.2 months, to obviously not reached as this is fairly early in that readout. And the median time to initial response was 6.6 weeks, so relatively fast. Median progression-free survival, 6.8 months. So this is smelling quite promising. Safety was the common side effects: acneiform dermatitis, paronychia, stomatitis, fatigue, hypoalbuminuria, wow, and 6% discontinued due to treatment-related adverse events. Michael, what's your take?
I like this study, and it's a fairly logical extension of amivantamab, and as you mentioned, any sort of regimen that can assist in the patients who are refractory after first-line therapy for head and neck cancer with, as you said, a fairly rapid response is going to be worth a look. I particularly liked that this was subcutaneous amivantamab because, as we have said before in episodes on Mariposa with amivantamab and lazertinib in the lung cancer space, amivantamab is notorious for infusion reactions and other toxicities. And it's of note here that 13% of patients did have administration reactions with the amivantamab, but they were all grade 1 to 2. So, and subcutaneous, a big problem that we had with the infusion was that, it's a weekly infusion, but obviously that takes time. So administration gets a tick. The efficacy, the preliminary efficacy gets a tick. I would like to see a little bit more data, particularly on overall survival, and maybe a head-to-head comparison with cetuximab, because as Josh mentioned, this is a small sort of single-arm study. So still a few holes to fill, but I think it's very, very encouraging.
That's it, Michael. Something in a much needed space. So next up is a breath of fresh air, or you might call it Wukong. I don't have anything to kind of link this to Wukong, so we're just going to say Wukong a lot.
I thought you were basically saying that Wukong translates to breath of fresh air, and I was going to be very impressed, Josh. You should have just run with it.
Yeah, sure, sure.
So yes, Wukong 28 is our next study, and it focuses on non-small cell lung cancer and EGFR exon 20 mutations, the troublesome middle child between the far more common exon 19 deletion and the L858R substitution. So these remain very difficult to treat. We've just come off a study talking about amivantamab. Amivantamab and lazertinib is actually approved in Australia for treatment of exon 20 mutant EGFR non-small cell lung cancer. But we're not talking about amivantamab and lazertinib in Wukong. We are talking about sunvozertinib, which is an anti-EGFR TKI specifically targeted towards exon 20 insertions, and has demonstrated anti-tumor activity across a broad spectrum of exon 20 insertion subtypes and other uncommon mutations. And it has been approved by the FDA in America for later line therapy on the basis of the previous Wukong 1B. So the Wukong 28 study, which is what we're talking about here, enrolled 324 patients with non-squamous, non-small cell lung cancer with EGFR exon 20 insertions. And notably, patients with brain mets were allowed if the metastases were stable after local therapy. So they were randomized 1-to-1 to receive sunvozertinib or chemotherapy, which in many places would probably be the standard of care, but the lack of comparison to amivantamab is something to flag with this one. And patients were stratified by the presence of brain metastases. The primary endpoint was progression-free survival, with the secondary endpoints being all of the usual stuff, focusing on overall survival and overall objective response rate. And here we have the results. So the median duration of treatment was 9.9 months for sunvozertinib and 7.6 months for chemotherapy. The median follow-up was 24 months for sunvozertinib and 18 months for chemotherapy, and the PFS was 10.3 compared to 7.5 months with a hazard ratio of 0.65, and this was statistically significant. The PFS at 12 months was 46 compared to 26%, so a significant improvement. The overall response rate was 58.9, almost 59% compared to 31.1%. The duration of response was 11.2 compared to 7.1 months. The overall survival data were immature, but numerically speaking, sunvozertinib is not numerically better than chemotherapy, 26.1 compared to 26.7 months. So that is something to watch. In terms of safety, the most common grade 3 or greater adverse events were elevated CK, diarrhea, anemia, and elevated lipase. So a few tricky biochemical toxicities there. So early days yet, but as you can see on the slide here, there's a significant improvement in changes to tumor size, some lovely waterfall plots, and the duration of response. But Josh, that is the Wukong 28, the breath of fresh air trial, as it shall forever be known. What do you think of this?
It's promising. Like, again, it's another really promising trial. And I think based on those waterfall plots alone, like, it's stuff that we need, more effective therapies for, especially in these sort of like resistant or, the non-standard EGFR mutation kind of cohorts of patients. So I think ultimately we will see this come more into the fore. I did note, the grade 3 adverse events, 20% elevated CK. What does that do with your renal function? Early days, I always say when it comes to tox, 300 patients, so not necessarily bad, but I think this will become, mainstream. That's kind of my take on it.
Yeah, I guess the question is, is it better than amivantamab and lazertinib?
Well, we might never know, because the thing
and we
is,
might
if
never
these are,
know.
so it will then come down to the question of sequencing and some real world data output, because there are people even with the traditional EGFR mutations where they've switched to second gen EGFR, and I have seen responses. That's anecdotal, of course, and I don't think there's a lot of evidence to support that. Get another huge lung cancer treater these days, but that is just one of those really unanswered questions, and the difficulties with these trials when they're designed somewhat simultaneously or a couple of years apart, and you don't know where to put them in the sequencing lane.
Yeah. So it'll be interesting. But next, Josh, we always do like in these sorts of episodes a potential novel target breaking away from the standard mold. So our next study is exactly that.
Yeah, well, this is an early study, Michael. It's a phase 1/2 study of JK06, not JK Rowling. Rowling, as you might think from Harry Potter. It's a 5T4 targeted antibody drug conjugate in patients with unresected, locally advanced, or metastatic cancer. So the background, this molecule, that's probably the important thing of this study, because it's a novel molecule. It's a tetravalent biparatopic DAR to MMAE antibody drug conjugate targeting the novel target of 5T4. So what is 5T4? You might be asking. It is not an investment company. It is a protein that is expressed by many different cancers, including lung cancer, breast cancer, ovarian cancer, endometrial cancer, a lot of cancers. There's more, but I'm not going to, harp on about that. And it's rarely in normal adult tissues, which is like, okay, is this one of those HER2 type receptors that you might see? So there's been multiple tumor-specific expansion cohorts in lung cancer, breast cancer, and also a basket cohort. This study itself enrolled patients with advanced, relapsed, refractory solid tumor, so good basket study, which I love. Unselected for patients with 5T4. You might be saying, well, why? We know lots of receptors. Nectin-4 is that classic one that, like, you have high, yes, you'll express, but you might still respond. So even if you have a low Nectin-4, we know that. And HER2 low is another prime example. This was a dose escalation had been completed, and they're currently enrolling tumor-specific cohort expansions. They were randomized to 2 doses to identify the recommended phase 2 dose in lung cancer and breast cancer. Fresh and archival tumor biopsies are being collected for retrospective correlation. I love that, of 5T4 expression with efficacy and safety. So it will give us a bit of more data on how we is it an intention to treat population? Is it a sub specific population we needed? Either way, let's go to the results, because that is what you are all wanting to know. 80 patients, overall 39 patients in dose escalation, 41 patients in the expansion cohort. The predominance of low-grade treatment-related adverse events, fatigue 35%, alopecia 18%, loss of appetite 18%, and a couple others including neuropathy and dry eyes. The grade 3 adverse events of interest, so gait disturbance, keratitis, and corneal ulcers. You do see that with some antibody drug conjugates, although it's rare. In the lung cohort, so 19 patients in this, the objective response rate was 32%, one with a complete response and some others with partial response. The longest continuing therapy was 51 weeks. So that's not bad, especially for an ADC. The responses were seen in both adenocarcinoma, squamous cell carcinoma, and EGFR mutant carcinoma. In the breast cancer, 7 patients, so not as respectable objective response, but 14%. Again, I say that when you've got 7 patients, you have to kind of take that with a grain of salt. This is a signal finding study, really. And the longest continuing therapy was greater than 27 weeks, which is the classic. There is always going to be someone who will respond to this therapy. Michael, what is your take?
Look, as I said, we always love a new target. And it's the unfortunate fact that a lot of these targets that show promise in the early phase setting, they don't quite cross the line when more information is gathered and we, find out more about the large-scale applications in certain types of cancer. But this is a early promising study, and I think, as you said, Josh, particularly in the lung cancer cohort, which was slightly bigger than the breast cancer cohort, and remembering that these are patients with refractory disease and really no appropriate available treatment options left. A response rate of 32% with one person having a complete response is nothing to turn your nose up at. And imagine being that person. You're basically told that you don't have much time left, and this is just sort of a Hail Mary, and you're more donating your time to science, as it were, and then boom, complete response. That must be incredible.
That's it. And I know we don't want to run late, but I just have the anecdotal story of my patient when I worked in Sydney, 7 years, phase 1 trial, breast cancer. No one else in the world got that far, and she was still just ongoing complete response. And this is the thing with these trials, really promising stuff. Michael, ESANOA, let's move on.
Yes, Josh is starting to crack the whip now. So ESANOA is another lung cancer study. You might be noticing a theme here. And it's another EGFR study. And it is a study of brain metastases specifically. So despite advances in anti-EGFR therapy, we've seen it with osimertinib and amivantamab, there's lots of stuff in there. Resistance to first-line EGFR TKIs represents a therapeutic challenge because for the longest time, we haven't really had anything after osimertinib. It's been chemo, it's been ABCP. From Empower 150, it's often been treatments that don't have very much impact. Specifically with regards to brain metastases, the cumulative risk of brain metastases occurs in up to 15% of patients at diagnosis and from between 29 to 60% over the first 3 years. And this is from a study that had a look at this, and you can see, just for those of you who are watching on YouTube, the incidence of overall synchronous and metachronous brain metastases, this is a meta-analysis of many different studies, is much higher across most cohorts in the EGFR mutant space compared to the EGFR wild type space. Similar sort of thing, not quite as dramatic with ALK. I should say EGFR is not quite as dramatic as ALK, but EGFR is notorious for causing brain metastases, either at diagnosis or down the track. So here's another mouthful of a drug, asanduterenib, which is a third-generation anti-EGFR TKI. And no previous trials have compared a third-gen TKI to osimertinib as monotherapy. And this is part of the reason I wanted to talk about ESANOA, because it is that study that we perhaps thought we wouldn't get. It is a comparison to osimertinib as monotherapy. We, of course, as we have mentioned a couple of times, have had Mariposa, which is the combination of amivantamab and lazertinib against osimertinib, but there hasn't been a one-to-one comparison of oral agents. The stratification factors for ESANOA were number of brain mets, less than 3 or greater than 3, and the EGFR mutation type. No weird mutations here. It was the exon 19 deletion and the L858R. They managed to enroll 224 patients into this randomized phase 2 open label trial with untreated EGFR mutant non-small cell lung cancer and stable brain metastases. And what they mean by stable is treated or asymptomatic. They were randomized 1-to-1 to receive either asanduterenib or osimertinib, and the primary endpoint was intracranial objective response rate and intracranial progression-free survival. And the results are quite interesting. So the median follow-up was around 19 months in both arms, and the intracranial overall response rate by blinded independent central review was 92.8 for, I'm going to try this again, asangiturninib versus 79% for osimertinib, which was statistically significant. The intracranial progression-free survival was not reached with the novel anti-EGFR compared to 17.5 months with osimertinib. And there was a trend in intracranial PFS across all subgroups. Moving out of the skull, out of the brain, the investigated overall response rate and PFS were consistent with the blinded independent central review assessments, and the median PFS of the whole body was not reached in the asangiturninib arm compared to 17 months in the osimertinib arm, which just snuck under the threshold for statistical significance at 0.047. Overall survival, as you would expect from these agents, remains immature. In terms of safety, the rates are pretty comparable. 99 compared to 95.6% of patients had some treatment-related adverse event of some severity. And treatment-related serious adverse events were slightly higher in the asangiturninib arm, which is 10.8 compared to 7.1%. So this is interesting, Josh, but I suspect you're chomping at the bit to say why it's ended up in this episode, Agents for the Future, rather than last night's episode, or the last episode, Agents for the Present.
Yeah. So I didn't know last night's episode was called that. So that's on me. I think, look, it's exciting, right? Because osimertinib is not that old in the scale of being approved, of which we've got a drug which is chomping at the bit, really seeing if it's going to be better. The things are, yes, the PFS is not reached in the intervention arm, but again, we don't have overall survival. And, we've been caught in this trap before where it's like, all right, maybe you're not actually going to become statistically significant. And that's something to kind of look at. But the objective response rate is an interesting one. Like you can see there's fairly significant penetration. The safety is the other thing. Treatment-related SAEs 10 versus 7%. Again, that's sort of not huge, but in real-world setting, will that actually have a significant impact? Unknown. But look, I think it's promising. Again, I don't think it's right there. I wouldn't be like, okay, use this drug as opposed to osimertinib in the first line.
Yeah, I think, and while the focus in this study is on the intracranial efficacy, it's important to note as well that osimertinib alone is no longer standard of care. We have FLAURA2, which is osimertinib plus chemo, which has demonstrated superior efficacy, but chemotherapy doesn't do a very good job of crossing the blood-brain barrier. So you may actually look at selecting this medication for someone who has high-volume brain mets diagnosis. There might be that level of precision
differentiation.
And differentiation, which would be great. But Josh, moving right along, why don't you try and decode the next study for us, because it is called ROSETTA.
I will. Look, we're running late now, Michael, so I'll try and speed through this. This is the ROSETTA Lung-02 trial, and this is a good one. I think, could this have been yesterday? Maybe. But either way, it's pumitamig. Just these names. Come on, guys, please, just a little bit easier. It's an investigational PD-L1 and VEGF-A bispecific antibody. It was designed to restore effector T-cell function, by acting within the tumor environment. This was a pre-specified interim analysis of dose optimization of this phase 2/3 Lung-02 ROSETTA trial, evaluating the recommended phase 3 dose. And who would have thought? I suspect this is going to go to a phase 3, but let's get there. Method, 44 patients were enrolled, so a small number. Untreatable or untreated advanced non-small cell lung cancer, no actionable genomic alterations, and a good performance status. And they were enrolled irrespective of their PD-L1 status. There were 2 simultaneous sub-studies divided into non-squamous and squamous, and about 50% each. And they were then randomized to 2 different doses of either 1,400 or 2,000 milligrams, and a histology-specific chemotherapy every 3 weeks. The primary endpoint was safety, objective response rate, and best percentage change from tumor size. Secondary endpoints, duration of response, disease control rate. Let's get to the results, the important thing. So this is the cool thing, right? Best overall response, complete response in 2 patients, partial response in 26 patients, and a stable disease in 12. And my maths isn't great, but 26 plus 12 plus 2 makes it about 40. So that's 40 out of 44, Michael. And so that's why I'm quite excited about this study. The objective response rate about 70% and the disease control rate about 100%. The duration of response was not mature at the time of cutoff, and the median best change in tumor volume was negative 38.2%. And look, I love that because ultimately, if this cancer doesn't grow, the patient can continue to live a good life. If it grows, it's bad. I say this all the time, but it's like, you can have a spot in your lung, but if it doesn't change, you can run your marathon, right? The PD-L1 level is available for 35 of the patients, and the majority were actually PD-L1 negative. Which is N equals 20, so maybe 50%. And that's an interesting one that, this whole debate about how do we target and select patients is becoming a little bit more murky, not just in breast, but in lung and a lot of our other cancers. And the median treatment duration was 4.5 months, and 30 patients, or 70%, were still on treatment at time of cutoff. The safety, 93% had an adverse event, greater than grade 3 was about 44% and were considered permitamig related in only 18%. And the discontinuation due to the drug was in 2 patients. So all very early. But my thing is this is promising, Michael. It's promising because it's sort of, yes, it's with chemotherapy, but is that sort of like an alternative or, a use of a previous drug like pembrolizumab or like another, not pembro, but like a PD-L1 using with another older agent to try and bring that together to make it more efficacious. And I love that.
I agree. And I also like that the efficacy was similar across lung cancer subtypes. There is still a little bit of a barrier, of a wall between adenocarcinoma and squamous cell, but pumitamig was efficacious for both. This is going to a phase 3. I believe it's coming to our centre in Brisbane fairly soon. So yeah, watch this space, pumitamig. It might be coming to a lung cancer specialist near you.
That's it, Michael. But why don't we take a travel somewhere down south? But we can look at colorectal cancer.
Yes, my great area of enthusiasm. I wouldn't say expertise, but enthusiasm. Yeah, so this next study is a study of a significant, I won't say untreated, but poorly treated, shall we say, cohort in the colorectal cancer. In fact, the majority, which is microsatellite stable colorectal cancer. We know about immunotherapy in the dMMR setting, but the pMMR or MSS setting, treatment hasn't changed significantly in many years. You could argue if you take away the biologics, the last major change came from the Mosaic trial all the way back in 2009, and that was the addition of oxaliplatin to 5-FU to give us FOLFOX. MSS colorectal cancers are resistant to immunotherapy, as we saw in the early data with pembrolizumab. But here comes Ivonescimab, which is not an immunotherapy per se or exactly, but it is a tetrameric bispecific, so another agent with multiple targets that targets PD-1 and VEGF. So think of it as a mushing together of pembrolizumab and bevacizumab. It's demonstrated efficacy and tolerability in phase 3 studies of non-small cell lung cancer. I'm assuming of adenocarcinoma because VEGF is really not good in squamous. So this study was a multi-part phase 2 study that evaluated Ivonescimab plus chemotherapy as first-line treatment for metastatic colorectal cancer. And this is the interim results for the multi-regional regimen extension phase of study. I'm not exactly sure what that means. Maybe they started in one part of the world and then have spread due to success. But they evaluated 2 doses of Ivonescimab, 20 milligrams per kilogram and 10 milligrams per kilogram in combination with FOLFOX. Again, a relatively small study, 49 patients. And all these patients had untreated metastatic colorectal cancer who had received or they could have received treatment for early stage disease as long as it was more than equal to 12 months prior to enrollment. They were randomized 1-to-1 to the 2 different dose levels, and everyone got FOLFOX for up to 8 cycles, followed by maintenance 5-FU and IVO until PD toxicity or withdrawal of consent. Pretty standard stuff. Interestingly, there was a note in the study that stated the maximum duration of treatment was 2 years. I'm not sure if they're sort of going like the immunotherapy route, where we sort of arbitrarily cut things off at 2 years, taking data from things like melanoma and cutaneous SCC. But I suspect it would be in practice, you would probably continue the 5-FU ongoing if someone had a prolonged response. And then you'd have a debate with the patient and amongst your colleagues about whether it's worth continuing the biologic. The primary endpoint was overall response rate and safety. The secondary endpoints were disease control rate and progression-free survival. So with a median follow-up of 7.5 months, so interim analysis emphasise that, the overall response rate was 70.8% at both dose levels, and all patients had partial responses, or all patients who responded had partial responses, no complete responses. The disease control rate was 100% at both dose levels, and the median progression-free survival was not reached. The 6-month PFS was 95% at the 20 milligram per kilogram dose and 84% at the 10 milligram per kilogram dose. So quite impressive, but sort of what you would expect for first-line therapy. It will be interesting to see how much better, if at all, this is over your standard FOLFOX Bev or FOLFOXATUX, FOLFOX Penny. Grade 3 or greater treatment-related adverse events occurred that were related to Ivonescimab to 40% of patients in the 20 milligram per kilogram group and 33% in the 10 milligram per kilogram group. Serious treatment-related adverse events in 24 and 16%. Treatment-related adverse events that were that led to discontinuation only in 4%. Versus 0%. It's important to note that these are of the Ivonescimab, not of the combination. And there were no grade 5 toxicities. So Josh, another promising agent in an area that hasn't gotten a lot of exciting, sexy stuff recently.
I do agree, and I also agree that this is early data. And whilst I just want it to be, add something that's going to really work, but again, it's not actually against first line standard of care anyway, because you're either going to do another EGFR or you're going to do Bev, right? Isn't that kind of the whole first line metastatic colorectal?
That is the paradigm, and I suspect
that's the paradigm, right?
If this is going to a phase 3, I would hope that would be the comparator.
I assume it will be, because all that stuff is off patent, so it should be. But Michael, time is of the essence. The finish is on the horizon. I'll jump across to Horizon CRC01.
Please do.
Great. So Horizon colorectal cancer, CRC01. So the background is, did we do this last night? No, maybe I'm just having deja vu.
You are having deja vu. We did do a Horizon study, but it was Horizon gastric.
There we go. So
don't worry,
I
haven't copied and pasted.
No, I'm sure you haven't. Either way, so it's the colorectal world, but it's a HER2 positive colorectal cancer, represents 5% of all cases. The anti-HER2 ADC that are standard of care in patients with HER2 positive breast and gastric cancer and have shown efficacy in multiple tumor types. This trial, Horizon CRC01, is assessing trastuzumab rezetecan versus the standard of care therapy. So 130 patients were enrolled. They had to be HER2 positive RAS-RAF wild-type colorectal cancer, had to progress after standard second-line therapy. They were randomized 2-to-1 to receive trastuzumab rezetecan or standard of care, being TAS-102, fruquintinib, or regorafenib. So late line. This is a late line study. I think you have to clarify that. They were randomized and stratified according to HER2 status, IHC 3+ versus IHC 2+-ish positive, and ECOG performance status 0 or 1. The primary endpoint, progression-free survival. Secondary endpoint, they snuck in an overall survival there, so I like that. That's always a positive thing. Let's look at the results. Hold your hats. We're going on an adventure. The demographic note, 70% of patients had HER2 IHC 3+, and 57% had a good performance status. I say ECOG 1 is still good performance status in my books, especially for a trial on colorectal that's third line. Like, that's fantastic. Median follow-up, short, 9.6 months. Progression-free survival, 5.5 versus 2.8 months, with a hazard ratio of 0.33. If we stop there, Michael, if you look at the, LONSURF data, we already know that data is more than 3 months because it was versus nothing. So an interesting thing when you look at PFS, again, maybe that's somewhat in real world settings. Maybe that was sort of not performing as well. That's an interesting question. Overall survival is not reached with a hazard ratio of 0.77. It is immature. And the objective response rate, 35 versus 2%. So that's good. That is something that you're like, all right, I am, my hat is curious. I am curious. My brain is curious. We're all curious. And the duration of response was 4.4 versus 3.4 months. And look, it's a smallish study. And finally, safety. Treatment-related adverse events was 48% for trastuzumab risatecan and 50% for standard of care patients. No patients discontinued treatment due to treatment-related adverse event, which I think is phenomenal that they haven't stopped. And there were treatment-related deaths reported in 2 patients with the drug, septic shock and myelosuppression. So I'm going to say probably in the context of having this drug, but also the caveat, these are heavily pre-treated patients. So my interpretation, Michael, is I'm curious. I'd like to see a large phase 3 study, larger than this, right? It's 120 patients. It's also a bit immature, but it is a promising study again.
I do want to draw particular attention to your pronunciation of immature there, Josh. That was a roller coaster in a single word.
No worry.
But yes, it's an interesting study. I think this is one of those areas, HER2 positive colorectal cancer, that is going to struggle to get beyond sort of small phase 2s just because it's so rare. Where I work, we've got a phase 3 study in the first line called Mountaineer. I can't remember exactly what the agent is, but all of the new diagnosis metastatic colorectal cancer patients we've been putting through for HER2, we have not found a single one. So they do exist, but I think it's going to be really hard. Of course, you in the background, you've got to mention TDXD in earlier line settings. I'm sure there are studies of TDXD that are bouncing around in this space. And so will trastuzumab risatecan come to the fore and steal TDXD's money? I'm not sure. I'm not really convinced, but it's always nice to have more ADCs.
That's it, Michael. It's nice.
And I am enjoying this circulation of ideas, Josh, which
well done. All right.
I'm proud of that one. That and the Rosetta one, I think I'm firing on all cylinders here. All 3 of them. So yes, the next study is called CIRCULATE. And this is an interesting study. I'm going to sort of be upfront. There are a number of issues with this study, but it is an interesting sort of signal finding, I think you said, in relation to an earlier study, Josh, study. So this is yet another colorectal cancer study, but it's of adjuvant chemotherapy. And this, if you are familiar with the Dynamic 2 study from Jeannie T down in Melbourne, you will probably be getting a bit of deja vu with this one. So the background adjuvant chemotherapy for stage 2 colorectal cancer is not beneficial in an unselected cohort. We know this. And there are some cases where you would consider chemotherapy, but usually that's for high-risk patients with T4 primaries or patients who have had perforations are the 2 main reasons you would consider chemotherapy. Dynamic 2 demonstrated non-inferiority in a CTD-guided decision-making matrix. So you can use the presence or absence of ctDNA to guide the benefit of chemo. It was non-inferior. However, they attempted to repeat that with stage 3 and they couldn't reach their primary endpoint of non-inferiority. So the ctDNA space is still very much evolving. CIRCULATE enrolled 1,396 patients, almost 1,400 patients. And they broke them down into patients who were ctDNA positive post-surgery, so ctDNA was only taken at a single time point. And they were randomized 2-to-1 to receive chemotherapy or observation. Chemotherapy was defined as capecitabine for 6 months alone or CAPOX for 3 to 6 months. And again, why would you give CAPOX for 6 months when the IDEAS protocol has demonstrated that 3 months of CAPOX is non-inferior to 6 months? I'm not sure. Patients who are ctDNA negative, so you have two groups, ctDNA positive and negative, were randomized 1-4, which is a very interesting randomization, to either observation on the study or off-study follow-up. So they were part of a sort of a long-term mortality follow-up, but they weren't included in the numbers. Patients and investigators were blinded to the ctDNA status, so the observation arm had both a combination of ctDNA positive and ctDNA negative patients. The primary endpoint was the disease-free survival in the ctDNA positive patients comparing chemotherapy to observation. So I hope that all makes sense because it is a bit confusing. Unfortunately, the trial ended early. This was an investigator instituted study, but unfortunately the funding expired, so it did have to end early. Now, I mentioned 1,400 patients were enrolled, but only 26 of these patients were randomized to chemotherapy, 287 to observation, and 1,083 to off-study follow-up. So while 1,400 as a raw number is impressive, the actual numbers of randomized patients were actually significantly lower. In the observation arm, only 15 patients were ctDNA positive and 272 patients were ctDNA negative. So in a similar fashion, you're having a very small number of patients who are ctDNA positive. In that observation arm, 3-year RFS was 87% for ctDNA negative patients versus 38% for ctDNA positive patients. So there is that signal similar to some of the subgroup analysis in Dynamic 2, that if you are ctDNA positive, your prognosis is significantly worse. The 3-year overall survival was numerically less, so 98% in the negative arm, 84% in the positive patients. And so we come to the per protocol analysis, and what they defined as the per protocol analysis. This excluded patients who did not start their assigned treatment, who either came off study before they started observation or didn't start chemotherapy. The 3-year recurrence rate in the ctDNA positive arm was 19% with chemotherapy versus 62% in the observation, with a hazard ratio of 0.23. And given the numbers, I really don't know how this achieved statistical significance, but it did, with a p-value of 0.009. The 3-year disease-free survival was 77% with chemotherapy and 38% with observation. Hazard ratio of 0.31, again statistically significant. However, the intention to treat analysis of the 3-year recurrence rate actually didn't meet statistical significance for its primary endpoint of 3-year recurrence rate, with a hazard ratio of 0.48, but the p-value was 0.075. So there's a lot sort of to break down there, and Josh, I'm sure you have many thoughts.
Isn't this the age-old question that we'd love to use ctDNA to guide therapy? That is the summary of this, and that, like, I know what they were trying to achieve, and I think if they had the money, we would have seen a really incredible trial. Just looking at that recurrence rate in ctDNA positive, you give chemo, you're going to reduce the likelihood that the cancer will come back. But then the intention to treat analysis, it wasn't what we needed. So, which is probably because they had such small numbers, right? It's kind of like intention to treat mathematical modeling. You need a big number of patients to actually figure that out. And I think that's probably the hard thing of this study is that I'd love to just, hey, ctDNA is high, give you chemo, we'll reduce the risk by 60%. Let's just say that's it. ctDNA low, you probably could omit chemotherapy. That's what we want. It's the same as in breast cancer or any other cancer space. If we can use a molecular profile to identify who to treat, everyone will be happy, good for the healthcare system, good for the patient, good for the clinician, good for everyone.
Yeah. I didn't write this in the notes, but also I remember reading something that the assay was developed something like 10 years ago. So this study has gone through a bit of a troubled gestation to get here. Funding issues, long-term development of the actual ctDNA assay. So it is interesting. It's laudable that they actually got the results out at all, really. But I think if we are using ctDNA to guide evidence, we do have Dynamic 2. We have this. So, in the stage 2 setting, you probably could use it if you have access to it. But at the same time, I'm not 100% sure, and they didn't go into too much detail, how much better it is compared to actual clinical assessment, because if you remember, there was no sort of investigated choice arm like there was in Dynamic 2. But we're almost there. Josh is trying to keep us to time. And our next study, we're going away from the colon and up into the liver with Emerald 3.
Yeah, that's it. That's it, Emerald 3. So this, we've spoken about a study very similar to this, or maybe this was the study several months ago, but this study is looking at embolization eligible hepatocellular carcinoma, specifically using the transarterial chemoembolization, which is the standard of care in this unresectable cohort. Then you've got the STRIDE protocol, which is single tremelimumab, regular interval durvalumab, which has shown overall survival at a 6-year follow-up. So we know that's, it's a standard of care from a systemic therapy option. This is a pre-planned analysis of TACE plus STRIDE plus or minus lenvatinib with TACE. I don't know if that's going to make sense, but it's a combination, right? The study was 293 patients. They were randomized 1:1:1 to have STRIDE, which is the immunotherapy arm, plus the TACE, which is the embolization, plus the lenvatinib as well, or just the STRIDE plus the TACE, or just the TACE alone. So 3 or 2 or 1, that's the comparison. The D&L continued for a maximum of 36 months or until disease progression, toxicity, withdrawal consent. They're stratified, lots of stratified factors. I won't go into it. The primary endpoint was progression-free survival of the SLT, which is that triplet arm, versus the single arm, which is always annoying. Like, you want to know what the doublet shows as well, because toxicity and efficacy, and we've had this conversation in multiple other trials. It's like, do you actually need the extra stuff? We don't know. When it comes to the endpoints, and this is where it gets somewhat interesting, the progression-free survival had a hazard ratio of 0.7. So what we know is that TACE alone versus TACE with treatment did better, and that was statistically significant. The overall survival, so that secondary endpoint, was a trend towards positivity with a hazard ratio of 0.84, but did cross that confidence interval. So again, not really no. When we look at progression-free survival of the doublet versus single, the hazard ratio was 0.71, and overall survival hazard ratio was 0.7. Again, not really sure. I don't think there might have been a sub-analysis. I don't have that in my notes here, but ultimately that wasn't what it was powered to do, but an interesting question there anyway. The 24-month overall survival rates was 66.9 versus 61% of triplet versus single. It's a little bit interesting because that's 5%, but we'd want to see sort of a longer term analysis to actually see if this is going to have an impact. And then 68 versus 57% of the doublet versus single. So that's probably a little bit more convincing. Safety, serious adverse events were 50% in the triplet, 36% in the doublet, and 15% in the single. As you'd expect, you're going to go down. You've got less drugs, less mechanisms kind of going through it. And leading to discontinuation, you saw 35% in the triplet arm and 20% in the doublet arm as well. So Michael, my analysis of this, which is, I think you just go 3 versus 2 versus 1. That's probably the only way to actually understand how this mechanism works, is that it is promising. And look, I suspect it'll probably end up being at least a little bit positive, but you have to kind of take that into effect of people that discontinue and the adverse effects profile.
Yeah, agreed. And look, we talked about tremelimumab and durvalumab yesterday in the previous episode, and we do know that that tremi in particular has a significant toxicity profile. We also mentioned in passing sort of the beacon pattern of things where a study will have a triplet, a doublet, and a single comparator. And it's actually the doublet that comes through as the winner. And looking at those numbers, Josh, I think that I can't really see a whole lot of numbers that are supportive of adding lenvatinib on top of STRIDE. And we know that lenvatinib has toxicity. And so
a lot.
Yeah, a huge amount. I mean, I don't think I've met a single patient who hasn't needed a dose reduction of lenvatinib at some point. So adding that onto an already toxic regimen plus TACE, you see the adverse events leading to discontinuation. You see the overall adverse events without numerically at least, a survival benefit. And again, as you said right at the top, we would love and we would prefer for there to be some sort of comparison between the triplet and the doublet. But numerically, I just wonder if this does become a standard of care, if this is going to be if it's going to be a doublet combination, so the durva, tremi, and TACE, as opposed to that plus lenvatinib.
Yeah, I think my only other point to that is to put the doublet as a secondary endpoint. And that is an interesting one. Like, I'm not a statistical wizard. Oh, no. I mean, because I think the thing is, like, they're probably wanting to see will Lenva help. But, like, it's just one of those things. It's like, I like the data. I agree with you. The numbers don't really fit to kind of lead to a triplet. And maybe there's, like, high risk cases that you might use a triplet if they're super fit, all those sorts of questions, or maybe you'll dose reduce the lenvatinib up front and maybe that changes this profile. We don't know. But we are trying to keep, Marco, you are definitely just the star of the show today, but we should move on. And you're not blue yet, but why don't we talk about BLUESTAR?
BLUESTAR, yes, let's talk about BLUESTAR. So this is another study focusing on gynecological cancers, specifically endometrial and ovarian cancer. Notably also presented at ASCO by Professor Linda Maleshkin, who is the head of Gyne-Onc down at Peter Mac. So it's nice to see Australia represented at ASCO. So the background to BLUESTAR is it is another study looking at a novel target, B7H4, which is a transmembrane glycoprotein that negatively regulates T cells, and it's highly expressed in endometrial and ovarian cancer. Puxi-SAM, which I'm sure has a much longer full name, but I've just written it down as Puxi-SAM here, is a B7H4-directed topoisomerase 1 inhibitor ADC. We love a topoisomerase ADC, Josh. They are growing at a rate not unlike the weeds in my garden. This has demonstrated promising anti-tumor activity in the dose escalation phase of BLUESTAR, and what was presented at ASCO 2026 was the updated safety and efficacy results. So in terms of recruitment, the authors recruited 93 patients with endometrial cancer, 78 patients with ovarian cancer. And they had to have relapsed or metastatic disease and an ECOG performance status of less than or equal to 1. And they had to be previously treated with at least one regimen that included a platinum, which stands to reason. There is some dosing specificities in this study. So EC, endometrial cancer patients, received either 2 or 2.4 grams IV every 3 weeks. OC, ovarian cancer patients, received 1.6 or 2.4 milligrams per kilogram IV every 3 weeks. And the reason I'm emphasizing that is because the results are stratified by those dose levels. The primary endpoint was safety and tolerability, and the secondary endpoints were anti-tumor activity per RECIST. So this is going to be a fact-finding safety mission, as it were. So we're going to be talking a lot about the treatment-related adverse events. And at the 2 dose levels for patients with EC, treatment-related adverse events were present in 92 and 88% of patients, respectively, for 2 and 2.4 milligrams per kilogram. 42 compared to 47% of patients had grade 3 or higher adverse events. The most common were neutropenia and anemia. In the ovarian cancer cohort, 87.9 compared to 97.8% had treatment-related adverse events at the 1.6 and 2.4 milligram per kilogram dose levels. 30.3 compared to 75.6 had grade 3 or greater treatment-related adverse events. And again, the most common were hematological neutropenia and anemia. 1 patient with endometrial cancer and 5 patients with ovarian cancer discontinued treatment due to treatment-related adverse events, which is not bad. In terms of the overall response rate, EC at the higher dose level was 48% and the duration of response was 7.1 months. At the lower dose rate, it was 34.1%. So numerically, the higher dose level is better, with not a huge difference in the level of treatment-related adverse events. In terms of the ovarian cancer cohort, 12.1% was the overall response rate in the 2 milligram compared to 24.4% in the 2.4 milligram per kilogram. So again, a significant difference, but you'll remember the treatment-related adverse events of grade 3 or higher in the ovarian cancer cohort was more than double for the higher dose level. So there will be some weighing up of toxicity versus efficacy. So we love a new target. We love a gynecological cancer study. We love an Australian study. So all areas that BLUESTAR really reaches into our hearts, Josh, but will be interesting to see if the B7H4 agonists and the ADCs, or antagonists, I should say, catch a bit of momentum.
I think, like many of these studies have said, time will tell, right? Time will tell. Should we move to our final study?
Yes, we are at the finish line, Josh. Let's see if we can get it under an hour.
Oh, yeah. Okay, so I got 4 minutes and 8 seconds left, guys. All right, so this is the GLP-1 agonist in breast cancer. This has kind of hit the news. Everyone's running with it. That's kind of the summary, right? It's not necessarily the thing we really need, but it's an interesting concept. So background obesity is linked to cancer. And also worse outcomes when it comes to breast cancer and those that are hormone receptor positive. The thought is partially due to adipose-derived estrogens and chronic inflammation, although this is not proven, it's hypothesized. I think that's the thing. Like, we think this is the case, but there's not like big randomized phase 3 trials that say, if your BMI is this, you will then get cancer. I think that's the caveat. And I think you'd agree with me, Michael, when I say that.
I would.
He's nodding. Yeah, good. I think it's just, people love to jump on bandwagons. I like to be like, well, this is the trial. So they investigated GLP-1 receptor agonist use in patients with resected breast cancer. The study was retrospective, so, not a phase 3, but it was analysis of data from the TriNet-X global collaborative network. It's like Skynet all over again. The methods, it was Michael's laughing. Propensity, thank you. The propensity score matched retrospective analysis of patients that are hormone receptor positive, HER2 negative metastatic breast cancer who'd received an AI plus a CDK4/6 plus a GLP-1 agonist versus an AI plus CDK4/6. So great. They're a high-risk cohort of patients. We know that. The cohorts were matched for age, race, BMI, heart failure, hypertension, type 2 diabetes, fulvestrant, and use of CDK4/6 used. The total number of matched pairs was 604, and the primary endpoint was overall survival. So kudos to them trying to do that. The results, the median follow-up was 15 months, so this is going to be immature without me even reading anymore. But so far, oh gosh, maybe it's not immature. The median overall survival was 67.9 months versus 49 months with a hazard ratio of 0.7. And if the p-value was 0.05, what they wanted, they did get it statistically significant. I think my caveat for this, though, Michael, is I think we need more data. Like, I know that's what it says, and maybe this is me being a conservative old man, but I'm like, I think there's no sub-analysis back. There's lots of unanswered questions. Obviously, it's actually going to be the answer. And the reason I say that is if you look at things like the Monarch-A trial, the Natalie trial, all of those adjuvant CDK4 trials, they did not read out with overall survival data at 18 months. So I'm trying to understand how they've got an overall survival that may be statistically significant here.
I think that there are a couple of ways to explain this. Number one is this is a retrospective analysis. I think they had something like 20,000 patients, most of whom did not receive a GLP-1 agonist. So that's why they only had 604. I say only. They had 604 matched pairs. Also, it's important to point out that the median of a data set is the middle value. So it's not
still only 30. So it's still only 30 months.
Well, no. My point is that it's not. It's the median value, as in the number that is in the middle, right? It's not the most common because that's the mean, and it's not. It's not the average. That's the mean, and it's not the most common individual value because that's the mode. It's just the number that happens to be in the middle. So the only way statistically that I could explain that is they had a bunch of patients who had very short follow-up because they progressed or died very quickly. And then they had a bunch of patients who were going for ages. And then the middle value was 15.8 months. That's the only way I can really explain that, because I agree, unless this is a heavily estimated overall survival value, those two numbers don't really match up.
I think, look, I do agree. And, now that we've sort of had this conversation and I've put my thinking hat back on after I lost it a minute ago.
It fell off your head.
It did fall off my head. Median overall survival. So they've obviously got more than 6-7 months. But I wonder if it's one of those things that you have a number of patients that are just living and living and living. So it's kind of like it's a skewed data set that's created this, and it's retrospective. So you need a prospective trial. Like that's going to be the next step. And look, it probably is worthwhile a prospective trial, but there's also so many other considerations here. People who had heart failure, people who had diabetes, who had hypertension. Are these all 65-year-old postmenopausal women who, are sitting duck to have a heart attack? And then you've got the 40-year-old who's got a GLP-1 and is like, well, yeah, I'm going to live for the next 30 years, irrespective.
Yeah, exactly. And I guess that's the other thing is the primary endpoint was overall survival as opposed to cancer-specific survival. So even though they did do an admirable job of trying to match patients, and we're focusing on matched pairs rather than individual patients, you are going to have people who die of, as you said, hypertension, cardiovascular, diabetic complications, and then people who are very young and otherwise fit and healthy who may or may not have a genetic predisposition, who then live for 1 million years. So it's difficult to assign the benefit purely to the GLP-1. And yesterday we had the Italian study on lifestyle alterations. How much of this suggested overall survival benefit could be achieved with lifestyle changes, aggressive lifestyle intervention that doesn't carry the risk of significant nausea, muscle wasting, or chronic pancreatitis, and a theoretical slight increase in the rate of pancreatic cancer. So it is interesting. It is hypothesis generating, but as the title of this episode suggests, it is not something that we would recommend today.
And I think my only final point, we are over time, is also I have patients who ask me about this, having a GLP-1. I'm always like, look, potentially. I think my thing is when it comes to tox profiles and you're just starting a treatment regimen, I always say don't put everything at the start, because who knows what's going to cause you problems. And if you get a side effect, but it's the GLP-1 versus the CDK, like it's that sort of grey zone. But Michael, let's stop. I will stop talking. Maybe you will too. Maybe we won't. Who knows?
Who knows? No, let's wrap that up because we have been going for more than an hour now. Dear listeners/viewers are probably bored to death. But we hope that you're not so bored to death that you won't join us for next episode, because next episode is the pointy end of this very abridged ASCO series. It is the big guns. It is the plenary. And we have a bunch of things. We've got lung cancer studies. We've got perioperative apalutamide for prostate cancer. And perhaps most impactfully, we have a study of abemaciclib in sarcoma. We love seeing rare cancers get a bit of a spotlight. So join us next time. We will be going through those studies in greater detail because they are obviously sat on a pedestal that is well deserved. But Josh, thank you, and we'll see you next time.
Bye.
Thank you for listening to this episode of Oncology for the Inquisitive Mind, produced by ADC Productions with the assistance of education grants from Pfizer, Gilead Pharmaceuticals, and Merck Pharmaceuticals. As always, these companies do not receive early previews or editorial input of any kind. The opinions expressed in this episode are our own, and the facts are, well, the facts. If you liked this episode, please consider subscribing to be among the first to hear our latest forays into the ever-changing world of medical oncology. You can find previous episodes of our show, including coverage of major conferences, interviews with world-renowned experts, and much more at inquisitiveonc.com. If you would like a particular subject covered on the show, drop us a line at inquisitiveonc@gmail.com. And if you happen to be in Australia, stay tuned for details about exciting in-person events we have planned for the near future. Thanks for listening, and we look forward to demystifying oncology again next time.